Thank you very much. So I'll join the other speakers in thanking the organisers for the invitation to this nice conference and to speak. And there we go. Right, so summary. Statement 1, no machine learning. So I'll thank the organisers and you can blame them. Okay, but on the other hand, what I want to do is, instead of, because I don't know anything about machine learning, I want to present an example, important question mark, of course question, I mean whether something's important is rather subjective, which I suspect could very much benefit from machine learning techniques. So I want to explain the problem and then maybe if somebody's interested. And basically the problem is exactly this: pattern feature recognition in ensembles of Gaussian PDFs. So, and when I say an ensemble, I mean millions or billions of them. They're multivariate Gaussian PDFs. And whenever I say a banded Gaussian, I mean that what I call the stiffness matrix, and statisticians would call the precision matrix, i.e. the inverse of the covariance matrix, is banded. And then the problem would be each of these millions or billions, they'd all be PDFs in a fixed dimension n for any one ensemble, and n would be of the order of a few hundred to a few thousand, like that. And the bandwidth of these guys is 42. So now, for fans of The Hitchhiker's Guide to the Galaxy, you know what the question is. Is everyone too young? All right. Okay, look it up on the web. Right. So how do we get these guys? So this is this bridge from molecular dynamics to bioinformatics. I'm going to generate these ensembles by marching along a genome. And so of course now genomes are published, and these Gaussians should be predicting the statistical mechanics. They should be some kind of equilibrium distribution of a short fragment of DNA embedded in a genome. And the point is, of course, there's a vast amount of information about bioinformatics, but they study patterns in the alphabet of letters. And I want to enhance that with mechanics. Right? You see, so the PDF will be something to do with the statistical mechanics, the physical properties of that fragment of DNA, which is something beyond knowing what the list of letters is in that fragment. And in particular, the question, it's not me that made this question up, lots of people think this is probably true, that consensus protein binding site sequences share common mechanical properties. And hopefully by the end of the talk, I'll explain what that bullet point means. Okay, so then, so I'm going to talk about the cgDNA. So CG stands for coarse-grained DNA. There's a family of models which I've developed. The dsDNA in the title is it's always double-stranded DNA. Okay, so I mean DNA also exists as a single strand, but we're not going to talk about that. So these models have a machine learning flavor in the sense that the models have, say, up to 20,000 parameters in them. And so that model, on the other hand, you have to predict PDFs for 4 to the n different sequences, where n could be a few tens. So you have to be able to model a whole bunch of different sequences with one set of parameters. So in that context, 20,000 is not so big. You know, because like if n is 10, that's basically a million, like that. But it certainly makes a lot of mathematicians, applied mathematicians, very unhappy when you say you have 20,000 parameters. And so I was happy to be able to add a joke, because this morning we learned that there's 2 types of elephants. Well, you probably knew it, but it was mentioned. So there's African elephants and Indian elephants. So John von Neumann famously said that if you give me 4 parameters, I'll model an elephant. And if you give me 5 parameters, I'll make its trunk wiggle. So he would need 10 parameters because he'd have to model an Indian elephant and an African elephant. But he might not be very happy about 20,000. But somehow computationally now, I mean, you'd never look at an individual value of any of those parameters, but you'd look at simple predictions of these models that have vast amount of parameters in them. So then, of course, there's an issue how you estimate all those parameters. And in the extremely unlikely event that there'll be time at the end, I'll say something about how we estimate it. I made some remarks on Monday about using Fisher information and natural gradient flow, and that's the context in which we use it, but I suspect I'm not going to get there. Okay, good. So then I'm going to make the assumption there's at least one person in the room who doesn't know DNA. There's some people in the room who know more about DNA than I do, I'm sure. But there we go. So what's the goal? So that red thing is a piece of DNA, and it's wiggling around, right? And it's being battered by a solvent. And what we want to do is we want to get an equilibrium probability distribution for the most likely state, which we call the ground state, and then the fluctuations, a measure of flexibility of this DNA as it's wiggling. And the point is that PDF should certainly depend upon the sequence along the DNA, and it should also depend upon this P, which is our parameter set. Now the point is that DNA functions in a solvent. The A of DNA is acid, and that means that it's charged, and so in fact it interacts strongly with the counterions. So you'd also certainly expect that that PDF depends, say, on the temperature of the solvent. It would depend upon the concentration of counterions. It would depend upon the species of counterions and all kinds of things. But that's all wrapped up in the P. Right? So the parameter set P is associated with one given solvent condition, and then you have to have, this is sort of the goal, right? So then I'll tell you a little bit about what the configuration coordinates are. That's a probability density function. It's a free energy. My definition of a free energy is something that you exponentiate with a minus sign and you get a probability density function. And then there's normalizing constants and stuff like that. And then this is a picture of a probability density function in R whatever. Okay. So then I have to tell you a little bit about what the W's are. So I'm not going to, I can't start from scratch from DNA, but I can give you the sort of backbone. Well, actually, that's a good joke. Another joke. The backbone. So DNA has two backbones, and they're anti-parallel, and they run that way, and then you run the sequence along one strand. Traditionally, we call that the Watson strand, and then the Crick strand runs in the opposite direction, and then basically the letters X, the standard alphabet would be four bases, T, A, C, and G, and then they'd be matched like this. So then T, A, C, and G look like this. This is the detailed chemistry of each of the bases. They then pair with hydrogen bonds like that, and we're going to coarse grain them. So the coarse graining is, there is a rule that if these coordinates are idealized, you stick them in a Lego brick, and there's 4 colors of Lego brick. And so then if you actually have an atomistic configuration like this, there's a rule where this yellow brick will go in here somewhere and then it turn there. So if you know an atomistic coordinate, there's some kind of fitting projection rule. That was a question, or you're putting your sweater on? Putting your sweater on, okay, good. Chilly atmosphere because the talk's so bad. Anyway, right. So that's the idea. So that's fine. And then we then have these rigid bases, and there's various conventions. Basically, so there's 2 bases are now rigid bodies, they're Lego bricks. So the degrees of freedom between a rigid body are 6, 3 translations, 3 rotations. They have to be called those names, and then the definitions are sort of the same, but not quite. These are called intra-base pairs because they're the 6 degrees of freedom between a pair of bases making a base pair. Then you end up with an average here. You also have 6 degrees of freedom when you go from one base pair to the next. These are the names of those, 3 translations, 3 rotations, and we call those inters. So then the coordinate vector looks like y, z, y, z, y, z, y, z, anything like that. Concretely, we use this particular code. My name is et al., but that's fine. So now I've told you vaguely what this W is. So the next assumption is we're going to assume that the U is actually a Gaussian. It surely isn't, but it's not such a bad approximation. And that's where we're going to start. So then, once we made that assumption for the free energy, the model is a rule to go from the sequence and the parameter set to the ground state mu, and go to the stiffness matrix K, which is symmetric, positive, definite, and banded. Okay? So we provide a bunch of different parameter sets to do this construction. I'll talk about that a little bit. Currently, each parameter set is estimated from a library of fine-grain, fully atomistic with the solvent, large-scale molecular dynamics simulations. Okay, so lots of people have worked on it. It's all on the web. You can look me up. Good. So we're in IPAM. So sometimes I have to give this talk to molecular biologists, and then they don't like this slide. But this is actually an interesting computation. Linear algebra is a cool thing. So A and B symmetric matrices, A and B invertible, and A and B vectors. Then if you sum two quadratic forms like that, it's a quadratic form. Okay? And so then, so you can write that as this, but of course this vanishes when x equals b, that vanishes when x equals a. If a isn't equal to b, this can't vanish, so you have to have a constant like that. But then what are the formulas? So you know you can compute c and c from capital A. So the formulas, you just add the matrices. That's just by comparing the quadratic coefficients. And the point of this slide is, the expression for the shift is C inverse times of AA plus BB. And in particular, the naked B never appears. It's always multiplied by B. And the naked A, little a, never appears. It's always multiplied by A. And that's just comparing the linear coefficients. So in particular, the overall shift involves the inversion of the matrix sum applied to these vectors. And the way we're going to apply that is, this is like a quadratic energy. So you should think about the x as being a set of coordinates, strains, and then there's going to be a rest state, a minimum energy state, and then A is going to be a stiffness matrix, and then that's an energy. But then a stiffness matrix times a displacement is like a force. I mean, there's rotations and stuff, so I'll put it in quotations. So the point is, to get the effective ground state of a sum of two quadratic energies, you actually average the forces, AA and BB, and then you compute an effective ground state displacement by doing that. Okay, so that's a triviality, but it's absolutely key in this DNA model, because now that was for two quadratic forms, but now what we do is we add a quadratic form for every junction. So you go along the DNA and you have a little quadratic form in each junction between four bases like that. And so then you have the superscript xy. That's a further assumption. It's saying the coefficient of this junction just depends upon the composition of that base and that base. Okay, so that's a locality. Well, there's two assumptions here. There's a locality in sequence, and there's a locality in the range of the force. The thing here is then that the basic variable is R18. Where does 18 come from? Well, there's 6 degrees of freedom there. That was an intra. There's 6 there, which is an inter, and then there's 6 there, which is an intra. So if you want the rigid body displacement from y to x bar, you need 1, 2, 3, 3 times 6, 18. So that's why this junction should be 18. So then the parameters then are, in fact, these are like the effective forces, if you like. They're the stiffness matrix times the ground state. And so then you can add up how many of the blocks and things are. And in this particular model, you end up with 1,500 parameters, 1,570 or something like that. Fine. So then we go on. Then what's the rule? So your parameter set now is you have 18 by 18 matrices. You allow the blocks at the end to be special. It allows fraying of the DNA. Then these are the vectors which allow you to compute the ground state. The standard alphabet is A, C, G, and T. We're actually going to allow an extended alphabet with M and N, and that will correspond to so-called epigenetic modifications of the bases, which I'll explain in a little bit. So what's the rule? So the rule is that you have all these little 18 by 18 blocks, and you add them up. And you add them up, and the point is they overlap, because so for example, this is the 2,3 junction. I haven't had enough to drink. My arm is shaking. Can I get a shot of whiskey? And then my arm will go. So the intra here participates both in the 1,2 junction and in the 2,3 junction, right? So that's why they're overlapping. Like that. And then the formula now is that the ground state then is the inverse of the effective K times the average of the vector, and the vector also overlaps because the intras participate in both junctions. And so the point is that according to these approximations, the stiffness matrix K, each block has local sequence dependence. And this vector sigma has local sequence dependence. But if you invert a banded matrix, it's dense. Moreover, its entries are non-locally defined. And so what you find is the ground state has non-local sequence dependence. And that's known to be very important in DNA. But it's quite curious. It's a bit confusing. There is a dinucleotide local parameter set which predicts non-local sequence dependence of the ground state. And that's exactly what's observed. Fine. So then I said epigenetic base modifications. So this is a cytosine, this is the atomistic detail, there's conventions, these are the corners are carbons, they're labeled around this way. So there's something called methylation, 5' methylation. You replace a hydrogen there by a carbon and 3 hydrogens. So that's an addition of 3 atoms, and you change a C to a methylated C, and that's what we denoted by M. There's lots of other epigenetic modifications, which I'm not going to talk about today. But the point is, if you open, never mind science and nature, if you open the newspapers, it's always epigenetic this, epigenetic that, da da da. It's this little piece of chemistry, and it completely changes how the DNA functions. Causes cancer, makes my hair go white, all kinds of things. My hair going well, yes, we'll make jokes about that. But I mean, so that's what epigenetics is, studying the consequences of modifications to the bases. It's maybe slightly simplistic, but we'll move on. Right, so that was the first thing I wanted you to know. So then the second thing I want you to know is sequence logos. So bioinformatics is just studying the strings of letters, right? So then there's things called transcription factors. CTCF is one of the transcription factors. It's a rather sexy one at the moment. There's then databases, and it's known that CTCF has a 19 base pair binding site, so it goes from 1, 19 is in there somewhere, and then you can average the sequence composition at each site. And so you then get something called a position weight matrix, and it says the probability of being A, T, C, or G at the 7th location in the 19 base is such and such. So then visually, instead of just saying the probabilities, they then weight the height by the base 2 entropy. So if you get to height 2, this would mean that C was 100% C, and if you get to a quarter, then the height would be 0, so there'd be no information. So this is a standard thing, sequence logos, and that's a particular one for this transcription factor binding site. Good. So what are we doing? So now the goal is I want to identify mechanically exceptional subsequences inside a chromosome. So chromosomes usually 10 to the 5th, 10 to the 8th base pairs, something like that. That's way beyond the scale. You'd never see naked DNA of that scale. So you never want to compute a PDF for a sequence that long. But instead, we want to compute this cgDNA local. So it's a marginal PDF for short subsequences Si in S. And now you can start to see where the ensemble of Gaussians come. So this is the PhD work of this student, Thomas Valen. And this is kind of a standard picture. This is what you can see in a light microscope and blah, blah, blah. But we're working down here at this level. And we're going to take, say, 10, maybe to 100 base pairs and compute a Gaussian, which is the mechanics of this subsequence. Okay, so how do you compute the marginal of a Gaussian? And it's now, as the American students would say, quiz time. Okay, so if you have a Gaussian, I'm going to give you the stiffness matrix, I'm going to give you the precision matrix, and then I'm going to tell you I want to marginalize over half the variables. What do? You compute the inverse, you then get a covariance matrix, and you take the submatrix corresponding to the variables you want, you invert the submatrix, and that gives you the stiffness matrix of your marginal. So that's dead standard, right? Now it's interesting, we have banded Gaussians. I have not actually found it said anywhere, but it's certainly true, that if you compute the marginal of a banded Gaussian, it's always banded. This isn't completely obvious, because when you invert a banded matrix, you get a dense matrix. You take a subblock of that dense matrix, and it has the property that its inverse is still banded. Yeah? So it's interesting. I mean, it's not hard, but it's very important for us because it means we can compute these marginals very, very fast. Right? But that's fine. And so then the stuff that the non-local ground state shape's very important that I explained. So that there's a technical point. So you actually you have a slightly strange marginal, but it's just on the sequence space. So you'd have a marginal for SI, given that you have a certain flanking sequence inside the big sequence, but that doesn't really matter. So what's the objective? So given a big long sequence, this is 10 to the 5, 10 to the 8th base pairs. Pick a fixed length. Up to you which length you pick. CTCF was a 19 base pair binding site, so you'd pick 19. I'm going to show you some examples for 10, but you pick a fixed length, then you want to locate sites, or that is subsequences SI of the given length with exceptional mechanical properties. And what does exceptional mean? So exceptional is going to mean that the PDF, this local PDF, is far from a sequenced average PDF for a fragment of the same length. So that's a particularly exceptional sequence, right, compared to, quote, average DNA. Question, how do you compare two probability distributions? How do you compute a sequenced average PDF? We've actually heard various discussions of this. Certainly comparing two PDFs, one and only one way, is the relative entropy, Kullback-Leibler divergence. That's the formula for it. But I guess most people here are familiar with that. And the key point for us is, we're going to do millions, if not tens and hundreds of millions of cases. Of course, there's a closed form, because that's a phase space integral, so that's a disaster, except when it's a Gaussian, there's a closed form. Actually, even evaluating the determinants and stuff, you have to worry about the linear algebra. But because it's a banded, you can do it. Okay, so you can evaluate that. So that's good. How do you compute averages? So there's many different notions of average PDF. In our parameter estimation, we estimate parameters by averaging over an ensemble of configurations. So the sensible way we average PDFs is we just make a super ensemble of all the configuration. I mean, if you have snapshots for sequence 1 and snapshots for sequence 2, the sensible way to estimate an average PDF is to merge the two ensembles and re-estimate. So that corresponds to this procedure. So you average the averages, and then you average the covariances, and then you get a new precision matrix. One small problem is that if you do that procedure where the Ks are banded, the inverses are not banded, and the average of the inverses of banded matrices is not banded. Right? So in fact, what we actually have to do is we get this average covariance whose inverse is not banded, and then we fit the best banded stiffness matrix to it. There's some cool linear algebra about that as well, which we might or might not get to. Okay, fine. So application. Mechanically exceptional DNA sequences made precise. Given a long sequence, boom, subsequence, boop. In the sense, compute now the Kullback-Leibler divergence of the local cgDNA PDF from the average banded, that's a banded average, not Bavaria. Right, so that's the marginal, that's the sequenced average cgDNA PDF. So that's now a well-defined mathematical question. And then what we're going to do is we're going to select SI with extremely high values of KL divergence. So one thing before I show you the nice result is, of course, that's an entropy, so it's non-dimensional, but you also need to scale it. So what we actually do is we talk about KL divergence per degree of freedom. And that's what I'm going to show you. So if you have a KL divergence of a 10 degree of freedom system or a 100 degree of freedom system, actually KL, well, let me not waste time with that. So what we do, if it's 10 degrees of freedom, we divide by 10. If it's 100, we divide by 100. So you just scale this unit of entropy by the number of degrees of freedom. And so now, then what we're going to do is we're going to identify the outliers, and we're going to see the sequence logos and see if there's a pattern. Great. So lots of time series analysis here. This is not a time series, this is a location series. So this guy is baker's yeast. It's actually one of the most studied genomes, because lots of people think it's very important for making bread. Personally, I think it's important because it's also brewer's yeast. That's the other reason to study it. It's the same thing, right? But it's very well studied. So it has 8 chromosomes. Here's 1. There are locations 1 through 2.2 times 10 to the 5. And then these are plots of the KL divergence of the window of length 10 from the average. One interesting thing is it never gets to 0. So there is no single point that's close to the average DNA. I mean, the minimum, that's 0.22, and then the height up here is 0.33. Right? So it looks like a time series, but it's not. It's a location series. But then you can do a histogram of the values, and it looks incredibly Gaussian. So then what we do is we can look at the sigma of that more or less Gaussian thing, and we call high outliers are all the guys higher than 3 sigma. All right? That's a standard notion of outliers, and those are the guys with the red crosses. Those are the guys that are beyond 3 sigma in this distribution. Now the question is, are they special sequences? Okay, so they are. And here we go. So the 3 sigma is also, by the way, we'll say this is Kullback-Leibler is bigger than 0.33. So it's almost entirely A's and T's. There's a few C's and G's there, but they're just never there. They're very well.
Sorry, is there an explanation why those outliers are just above and not?
Because they're high outliers. So I'm just looking at high outliers. Sorry, did I say that? I think I said far from the mean. I should have said that. Extreme high values. So I'm just, yeah. I've taken the slides out for length, but you can plot low outliers too. But in some sense, the low outliers are slightly strange. The low outliers are the sequences closest to average. See, in this sequence, I mean, average should be 0. And it's like you're all lying on a sphere in high dimension. Yeah, but it's certainly true. You can do that. I just don't have the slide. Yeah, but good. So then there's a sequence logo, and it's quite remarkable. All you get is A's and T's. So to my knowledge, that was never known before. Now, whether it's interesting is another question, but that's the kind of thing. Right? So the point is, you then have this statistical mechanics localized PDF, and the things that are furthest from average are, essentially only have A and T base pairs. All right. Okay. So is there anything special about the particular chromosome 1? No. It's the same for all 8 chromosomes. Is there anything special about 10 base pairs? Well, it works for 11 base pairs. It works for 20 base pairs. It works for 147 base pairs. This is the test, if you've ever read it. This is the number of base pairs wrapped around the nucleosome. Like that. But it's clear that there's something very, very special about all these guys. Right? So that's nice. Now, if I didn't go too fast, the cgDNA PDF depends upon dimer steps. So it's not actually enough to know the frequency of each base. You need to know the frequencies of each base pair step, right? Because it was a KXY. And so now you can now look at the dinucleotide step sequence logos. So now, in fact, there's 16 possibilities, 4 times 4. And all you see are green is you get AAs and TTs. In fact, you can't tell the difference between those because it's the 2 strands. But really, all these high outliers are sequences with lots of AA steps, which is the same as lots of TT steps, because you don't know which stand you're on. So that's quite remarkable. So then you can ask, okay, are biological chromosomes very different from random chromosomes? So then we can just make up a random sequence with quarter, a quarter, a quarter. And again, it sort of looks the same, and you have a fairly nice one-dimensional normal distribution. The outliers now, it's 0.29 instead of 0.33, like that, so it's lower. And then actually there's more of them. And the sequence logo is slightly less clean. It gets polluted with some other sequences. So it seems as though the biological sequence actually has more AA steps and TT steps than you'd expect, right? Which is also nice. So now we then talked about epigenetics. So the thing is, so we're now going to say, okay, I want to see what happens if I methylate C's in CPG steps. So I'm going to take the random sequence, I'm going to keep the same average PDF without methylation, but now I'm going to methylate every single C in every CPG step. So now, this is the histogram. I think we agree. That's not Gaussian, right? And in particular, so we don't talk about standard deviations, but now we define the outliers to be 99.7 percentile. But now the critical value of Kullback-Leibler is 0.47, so way higher, okay? And then you can say, and this is qualitatively different. So what's going on? Well, this is, actually I think I didn't write it, this is still 10 base pairs. So what's the simplest possible explanation? Well, the simplest possible explanation is that is actually a Gaussian where there happens to be no methylated steps. And that's exactly the Gaussian we saw before. But then as soon as you methylate one step, you get much farther from average. And if you methylate two steps, you get further from average. And then there's just a tiny little bit of 3, and in a 10 base pair, it turns out you can't have 4 methylation steps. So this, making this methylation, which is 3 atoms on a C base, just completely wipes, it's incredibly strong statistical mechanic perturbation. So that's interesting. So now we have the dimer steps. Actually, I'm a co-author of Ilya Harenko, and this was Ilya Harenko's slide. Why? It's his flag. It's Ukrainian. See? It's blue and yellow. But anyway, never mind. Maybe there's somebody else Ukrainian. But I think that's right. I think it's Ukrainian. And yeah. So the outliers now are no longer the AA steps. They're the methylated guys. And it's a strong indication that methylation, which is known to be biologically and medically very important, it's very important in cancer, has a strong effect on the statistical mechanics. And it could be, it's how it functions. Good. So that's one thing. So that was asking what sequences are furthest from an average sequence. But now I can ask a different question, which is I can ask for sites that are mechanically close to a particular sequence motif. So then back, this is my Jasper database CTCF transcription factor binding site. And so it appears in this case, there's 930 fragments, each of which is about 200 base pairs. Each is containing one experimentally known CTCF binding site. So it's there, etc. And then you can ask the question, can we identify, because the sequences aren't the same, and then the question is, do those sequences have something physically in common that's not immediately obvious from the sequence? So the way we do that is, well, I'll show you the data in a minute, but the other thing is we have this PDF averaging routine. So I could average over any ensemble. So I'm going to average over the ensemble, which is all the binding site sequences. So that will give us a consensus cgDNA lock PDF for a CTCF binding site. Yeah, fine. So now what I'm going to do now is I'm going to pick one of the 930 fragments, and then I'm going to run the cgDNA PDF for each of the 930 binding sites along that fragment. So this is 930 curves, right? Bunch of locations, and you get a dip. Guess where the binding site is? Right, has to be there. And it has to actually go to 0 for one sequence, because you run the binding site that appears in that actual fragment, and that has to hit KL0. But the point is that lots of other ones, the other binding sites, which aren't identical sequences, similarly have very low KL divergences. So this is sort of an indication that something's good going on. The statistical mechanics of the binding site isn't so bad. So now this projector has an interesting color map. This is usually cyan, and it usually screams out at you, but it's not so obvious. You see this light blue curve? That's the average guy, and it really dips a lot. So that's good news. The bad news is that this image is a blow-up of here, and there's black dots here. So that means one of the binding sites appearing in this database is in fact far from the PDF of the binding site in this fragment. So that's one plot with 930 curves, but it's like a matrix of interactions. So I could give you 930 such plots. They always have dips at the binding site, but it's not entirely convincing. There's all these dips here, and it's not exactly clear what's going on. So it's not entirely convincing. So there's various things that could have gone wrong here. So perhaps the potentials in the MD simulations are not sufficiently accurate. Maybe the multivalent ions are important, whatever. Any kinds of things could be going wrong. We can check that the cgDNA predictions of shape and stiffness, if you believe the MD predictions, there's no error there. But the interesting thing is this, well, interesting question mark, subjective. The interesting thing to me is, and I think to others who work in binding sites, is that it's too strong a condition to ask that the Kullback-Leibler divergence is small. And the reason is, it's not a metric, of course, but it vanishes if and only if the PDFs are identical. So that means every eigenvector and every eigenvalue have to be identical. And in particular, the eigenvectors with large eigenvalues have to be identical, and that can't be physically pertinent, because if it's a binding site, it's going to be a low energy mode. The thing that you really want is that the eigenvectors for some number of the small eigenvalues are close. So that's easy to say. It's physically sensible, but it's not easy to make precise. And that's where I think it would be very interesting to try and use machine learning to try and learn what are the common features in the PDFs for binding site sequences versus PDFs for not binding site sequences. And mathematically, it's just going to say, so CTCF is one protein. There's databases for hundreds of these proteins. So for each protein, you'll get an ensemble of Gaussians. I'll tell you this subset of the ensemble are binders, this subset of the ensembles are not. Please identify what are the features of the stiffness matrix and the ground state that's in common in the binders. And so basically, that's why, in fact, we have a project with Christoph Schutter in Berlin that we're planning on doing that. But the statement is, I know nothing about machine learning. I'd be interested to know if people are interested in that, we could talk about it. Have people applied machine learning to look at patterns of Gaussians? I personally am unaware of this. If people know of literature on that, I can't imagine that they've done it for molecular biology applications, but I'd be interested to know if there's people who've classified similarity patterns between Gaussians, but it cannot be full Kullback. It can't be a full Kullback-Leibler. It can't be a full divergence because that's too strong, right? You want to have something. You have a set that has property X, a set that has property not X. What's common for the X's, right? That went a lot faster than I thought it would. Okay, good. So it's 50 plus questions, right? Yeah. But you said it too late. You didn't say it before. It's okay. I'm ahead. It's fine. So yeah. So actually, that's where I wanted to get to, right? So now I said previously. Well, so now I want to just say a little bit about this. And in fact, this will also, I think this is of more general interest, even if you don't care about DNA. This is a more detailed picture of a piece of double-stranded DNA. This is 5' this is one orientation anti-parallel. Here are the bases, yada, yada, yada. So the data I showed you is actually for the cgDNA, which is the rigid base model. So the phosphates are in the model, but only implicitly, right? Because you're looking at this, in fact, it's a marginal. So now cgDNA plus, which is what I'm going to talk about, is going to add degrees of freedom to every phosphate group. And so that's an inter, that's an intra, and then we're going to ask another 6 degrees of freedom for the rigid body motion of this phosphate from that base. And now what happens is, instead of having a level as being 2 bases, a level is 2 phosphates and 2 bases. And now, so now it's important. I think we already mentioned this notion of marginals. So now I could try and derive a model that only has the inters, or I could derive a model which has intras and inters, or I could try and derive a model that has the phosphate coordinates and the intras and the inters. And the point is that whenever I have a more detailed model, it's a nested chain. So the degrees of freedom in the inter model is a subset of the rigid base model, and those degrees of freedom are a subset of the degrees of freedom of phosphates and bases. So you can actually take a chain of marginals. So now I've told you that we estimate these guys by running MD simulations. So the first thing we do is we run a few long duration MD simulations, and then we can estimate averages, which I'm not going to show you. Oh, I will a little bit actually. But we can then estimate centered covariances along the time series. So if I just observe the inter degrees of freedom, I get a covariance matrix, and I invert it, and I think we can agree that doesn't look very close to banded. Bad idea, right? So there are, so a nearest neighbor rigid base pair model would be these block diagonal 6x6 matrices, and it just doesn't fit with the MD at all. So the cgDNA model looks like this. So now I have more degrees of freedom. I have inters and intras. I have a bigger covariance matrix. So I've made it a finer grain model, have a bigger covariance matrix, but then when I invert it, it's much closer to being banded. Right? And so the sparsity, I showed you this 18 by 18 sparsity pattern, and there are entries outside that sparsity pattern, but they're not so big. Right? So now the question is, you want to ask, okay, so is that model good enough? So what you then do is you add the phosphates. You now have an even bigger covariance matrix. I then invert it, and hey presto, I mean, that's not fudged. Right? So, I mean, the MD data is screaming at you that you want to put phosphates in, and then the stiffness matrix is almost completely banded. There's these tiny, tiny entries outside. Now, the other striking thing about that data is you see these horizontal and vertical white lines. So you have to worry about scaling, but this is a sensible scaling, and those white lines are real. And what they say is, well, it's an interesting question. Degree of freedom 1 and degree of freedom 2 are not coupled. What does that mean? Are they correlated or what, right? So I say coupled if there's a non-zero entry in the stiffness matrix, and they're correlated if there's a non-zero entry in the covariance, which is the inverse. So the phosphates on the Crick and Watson strands are very definitely correlated, but they're not coupled. There's no direct energetic, that's what these bands say. These bands say there's no direct energetics between any phosphate on the Watson strand and any phosphate on the Crick strand. And that's not an assumption, that's just like the data just screams at you. So yeah, so this is the cgDNA plus model. So now this is the 42 by 42 blocks, but it's the same pattern. You just have overlapping blocks. It's the linear algebra. You get non-local sequence dependence. You do all this stuff. That model has the 20,000 parameters in it. Okay, so this would make John von Neumann unhappy, probably. Certainly makes lots of referees unhappy. Presumably it's not John von Neumann anymore. So you do all the same stuff, like that. And so now, this is now a comparison between an atomistic MD simulation of a 24-mer fragment that is not in the training set. I repeat, not in the training set. And then we compare it with the predictions from the model. So, in the front row, can you see the difference? Maybe a little bit there. Back row, you probably can't see the difference. And then you see the scales. That is 0.01 of an angstrom. And the signal is like vast, right? So I declare this as, can't do better than that. I mean, I'm not going to do any better than that. And this is the inters, this is the intras, these are the phosphates, da 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 da. And the other statement is that, I mean, people who know about DNA, they get very unhappy whenever you say you have a dinucleotide set of parameters because they know there are non-local sequence dependence. But this is now, this is actually too small. So this is an alternating AT sequence, and then there's one site where I change a T to an A. So it's called, I mean, you could call it a SNP, single nucleotide permutation. So you change the base composition at one site. And these are visualization of the two ground states. And the two ground states could change completely non-locally. So this linear algebra, hook and crook or whatever, exactly matches the MD. And I can make, no, one more slide, right? So you can repeat everything I did about sequence logos and stuff like that. It's all the same, if anything, right? Except if we remember the famous, this plot, right, about matching to sequence logos. So now, I guess you guys, like me, all use TeX, right? So then you have to load the file name in to get the figure. So I have nightmares over this figure to see if I've loaded the same image twice. But I haven't. There's little differences, right? And so you get this really, really accurate, I mean, this is as good as the molecular dynamics coarse-grain model, and the signal of the Kullback-Leibler divergences doesn't get any better. Right? And so, actually also doesn't get any worse, interestingly enough. It's quite remarkable. And so that's why that's why I intend to try and use machine learning to try and understand what is going on with those consensus binding sites. And it would probably go a lot faster if I could find a collaborator who knew any machine learning. And after that, I will say thank you.